Thank you for the introduction and for the invitation to speak here. It's a privilege. Would have been even nicer to be there, but obviously, for this time, it's fine to have it remotely. So yeah, hello everyone. So I hope you hear me well, and thank you all for coming back. Actually, so yes, arranging times is always a little bit tricky, but yeah, happy that you managed to come back, and thank you for that. So, can I just see whether you see my presentation okay?
Yes, yes,
Excellent.
we can
Very
see that.
very good. Thank you. So today I'm going to tell you about topologically active polymers, which and in particular I will focus on. So this is the name of my group, and so I'll tell you perhaps by the end you see what I mean with topologically active polymers. So my group is funded by the European Research Council, ERC, and the Royal Society, and recently we received some more funding from Leverhulme to do the machine learning part of that I will tell you about the second part of this talk. And just as a mean of kind of introduction of why am I doing what we do is I want to tell you a little bit how I got here. So maybe I can start by telling you that so when I moved from Warwick to Edinburgh to do my postdoc, so I moved from studying polymers. So I was doing polymer physics during my PhD. I started to do more genome organization. So try to understand how DNA is organized inside cells using polymer physics. And this is one of the first images that I saw when I was catching up with the literature, and this is a reconstruction of the genome of our genome in a human genome in a human cell, a HeLa cell, a cancer cell. And what I want you to take from this image is that it's very entangled. It's very packed, right? So this DNA is very packed. So we have actually 2 meters of DNA in each cell. So in each of our cells, we have 2 meters of DNA. So which is, if you scale it up, it's the equivalent of packing 20 kilometers of rope, or a very thin rope, of a hair thin rope inside your cell phone. So this is how smart nature is in packing the DNA inside each cell. So if you take all the cells in your body, which is a trillion cells in your body, then you multiply 2 meters by trillion, so you get to the end of the solar system many times with only the DNA that you have in one body. So this is how much DNA we contain. And so this kind of when I realized this, I kind of to me it was very impressive that in spite of this extreme packaging and entanglement, the cells could still operate, do gene expression, but also replication, for instance. So here you see a little movie of two cells that split their DNA and divide. So the DNA is in purple. So you see the purple bit are kind of condensed and then split into the two daughter cells and then decondensed. And in each of that cell there is 2 meters of DNA. To me it was mind blowing, right? So how can you manage all that entanglement? Because if you think about in terms of materials, of polymer materials, for instance, it's very difficult to manage all those entanglements, to actively remove the entanglements, being able to separate polymers into, let's say, wherever you want, and then put them back where they were, so kind of re-entangle them back. So this is something that fascinates me, and this is why kind of my group focuses on two things. One is to understand how cells manage these entanglements, and manage topology as well. There is a lot of topology involved. We will see it in a second, perhaps, and also try to understand, kind of take inspiration from how nature does it to make materials. So to kind of almost like export some of the technology, let's say, that nature has evolved in terms of using DNA and using proteins to perhaps make materials. And so this is a little bit of an overview of what my group is doing. So we look at topological viscoelasticity of DNA fluids. So for instance, these are just an example of simulations of supercoiled DNA models. So inside this box, I've done, this is a molecular dynamic simulation of supercoiled polymers, so polymers that can be twisted up and form, and they're not just simple linear polymers, but they're circular, and they can also be twisted up and coiled into themselves, so almost like DNA does. And you can study, for instance, how they thread. So there is quite a group of people interested in how ring polymers thread through each other, for instance, and how this affects the material properties of complex fluids. We did also experiments using DNA and restriction enzymes. So here, for instance, we studied how the viscosity changes as a function. So if you have a bulk of entangled DNA and you add restriction enzyme, which is an enzyme that cuts DNA, like ones that you find in bacteria, for instance, or even in our cells, and it starts to cut DNA, we were studying how the viscosity changed over time. We did, sorry for this picture, I'm not sure why it comes up like that. We did some, so this was during my postdoc. I was looking at molecular dynamic simulations of how HIV integrated, so the DNA of HIV integrated into the genome of human cells. So in this case, I was looking at, so this is an example of DNA in gray and orange, represented as bead spring polymer model, wrapped around these blue spheres, which represent histones, which are proteins that help package the DNA inside our cells. And then there is this green ring here, which represents the circular viral DNA that tries to integrate inside the genome. And we were studying where this integration happens, and found that the regions of near nucleosomes, near histones, where the DNA is pre-stressed, where there is a lot of bending stress, were the favorite places for the viral genome to integrate. And indeed, then we collaborated with the experimentalists, who can map where the viral genome can integrate in the human genome, and we actually found correlations between the pre-stressed states and the integration sites. This at the bottom is another example of how we essentially I come up with a recolorable polymer model. So this is again a polymer in terms of coarse grain representation of chromatin, in this case of our genome, and we know that in our genome there are regions that are active, in the sense that their gene is expressed. And these are in red, for instance, or blue, and they can infect each other. So the regions that are silenced, where the genes are not being expressed, they can infect the ones that are active, and vice versa. And then in some conditions you can find this stable states or metastable states, in which you have coexistence of silence, blue, and active red states. So this was quite interesting work during my postdoc. And then it started to kind of move towards more DNA materials. So one of the most interesting ones that I found so far is this kinetoplast DNA. So this is a genome from a parasite. So this is a blood-dwelling parasite called trypanosome. You can extract the genome from the mitochondrion, and we put it on a surface and looked at it with AFM. And what we saw is that it's made of interlinked DNA rings, small interlinked DNA rings. And so we studied the topology of this, so how these rings are interlinked and so on. So I will not tell you about this story, but if you're interested, I can tell you later. I can give you references for later. This is a very interesting material as well, because if you think about this is a 2D, it's a two-dimensional, when it's put on a surface, this is really a two-dimensional sheet, okay, made of interlinked DNA rings. So which is very difficult if you want to make it, for instance, synthetically. This is very difficult. This is the type of stuff that Professor, for instance, David Lee will do, you know, with the molecules. But those are the molecular knots, for instance. But these are, or the weavings. But this is much more scale. This DNA is quite big. So this structure is microns inside. So this is pretty big. So I think if we were able to recreate in somehow how these parasites can assemble these structures, we could make membranes made of these Olympic networks, so networks of interlinked DNA rings, which I think they have interesting properties. So one property that we think they have is that they are ultra soft. So they have a softness that is 1,000,000 times lower than that of lipid membranes. Well, I've also worked on topological glasses. Maybe this is this is also interesting. It's a kind of it's also, it carries on from my PhD. Is where we study a dense solution, for instance, of ring polymers or rings mixed with linear or linear and or these are we call tadpole polymers, so linear and rings fused together. And these were actually made with polystyrene by a Japanese group, and they're quite interesting because then what happens is that these tails can thread through the head of these tadpoles, and then if the tail becomes long enough, then you have so many constraints that the tadpole itself that is threaded it cannot relax their conformation, but that tadpole that is threaded is also threading another one, and so there is a proliferation of constraints which eventually create a glass, which is not microscopic glass. It's not microscopic and it's not a temperature glass, so it's not lowering the temperature, but it's changing the let's say the conditions of using topology in some sense to create the conditions for which the macroscopic polymers cannot really diffuse anymore. And then there is a new kind of branch of research in my group where we started to use machine learning. We looked at knots, but we are starting also to look at molecules. So here is kind of, we were using diffusion models to take fragments of a drug molecule and simulate many different possible candidates for new drug molecules that connect these fragments inside a protein pocket. And we are targeting in particular topoisomerase. Sorry, I moved a bit quickly. So that was kind of an overview of what we do, and today I would like to kind of focus on two projects. One is topological viscoelasticity, and the other one is the machine learning of knots. I was undecided which one to tell you about, and I wasn't sure which one you will find more interesting. So I'm going to try to squeeze both in. So the first one was led by Yair Fosado, who actually spent a few years in Japan with Professor Takahiro Sakaue in Oyama Gakuin, I think. And this is Georgia Palombo, a PhD student who graduated just yesterday, actually had her viva. So essentially, this project relies on a system made of DNA, and the DNA system, this is quite important that to understand kind of the design system. The design system is made with this DNA nanostar. I'm going to call this a DNA nanostar. So it's made with 3 single strands of DNA. So this I assume that you guys know that you can take a single strand of DNA and you have a series of nucleotides, which can be A, C, G, T, and you can design any sequence that you want, essentially. And then what you can do is you can design 3 sequences so that they are complementary in this way. So for instance, you have strand 1 that goes from 5' to 3' here. Then strand 2 is complementary to the second part of strand 1, and then it's free here. And then you design a third strand that is complementary to this part of the second strand and the first part of the first strand. I hope I get the message across. And then this part here is kind of, it's a bubble. It's open because there are unpaired nucleotides, so they don't pair up. At the end of these sequences, there are also sticky ends, so there are unpaired nucleotides. Okay. And so what happens is that if we lower the temperature of the system at low enough temperature, what happens is that 2 sticky ends, because they are self-complementary, they will stick together. Okay, and below this Tm2, which is the melting temperature for the sticky end, that sticky end, that connection between 2 stars is relatively stable. So we're not the first to study this. So Omar Saleh, Francesco Sciortino, Eric Heiser, Takinoue also studied this system. So what we know, and other people also have found, is that there is a phase separation region. So as a function of DNA concentration, a phase separation region means that you form these droplets where there is DNA rich inside and just water outside. But then if you push this to, and this has to be a low enough temperature as well, but if you push it to high enough concentrations, at some point you get a gel. And this is really a hydrogel, something that you can make in the lab. So this is what Georgia was making, and yeah. So this is something. This is a region of the phase space that um, um, it's not very well studied typically. So typically a lot of people study this region here within the phase separation where you have these droplets. But we are interested in this phase here. So the way we make it is essentially by ordering these three oligos separately. Then we mix them in equimolar quantities and we ramp from very high temperature to from 90 degrees. We slowly cool it so that first the stars are forming and then the stars start to join into a network. So the first thing that we did when we got this hydrogel was, well, we can do two things. We can either do bulk rheology or microrheology on it and study its we wanted to study essentially its mechanical and viscoelastic properties as a function of the design of DNA nanostar, because you can change the design. You can have 4 arms, you can have longer arms, you can have shorter arms. And so this is what we did. So these are curves from bulk rheology. Maybe just a word for those of you who don't know bulk rheology. You put your sample in between two plates and you rotate one plate with respect to the other. And what you do, you measure the strain, so how far you've moved the plate, and the stress, so how strong, how much force you have to put on the plate to rotate it to apply a certain strain. And so you have this strain, you can have essentially strain stress curves that tell you how much strain you put in the sample given a certain stress. But you can also do this oscillatory rheology, which is shown here, which essentially shows, so these are curves at different temperatures. So let's take for instance the, I think it's the other, no, sorry, this is a concentration, sorry. So let's take this red curve here, for instance. So what these curves are showing is that if I rotate my plate a frequency of 1 Hertz, okay, so 1 spin a second at a certain amplitude. Typically, it's a small amplitude to keep the material not too disturbed. I get so I measure a stress of, let's say, 1 kilopascal. Okay, and this stress is split into an elastic and a viscous component. So the G prime is what measures the elasticity, and the G double prime is what measures the viscosity, the plastic response rather than the elastic response. So at frequencies, if I spin my plate at higher frequency, so at 10 Hertz, 10 times per second, then what I see is that this solid curve, which represents G prime, is higher than G double prime. And this means that the hydrogel responds more elastically and it's less viscous. So it's dominated by elasticity. On the other end of the scale, though, if I move my plate more slowly, so 0.1 Hertz, so only once every 10 seconds, the system is dominated by viscosity, so by this dashed line, and elasticity is less important. What this is telling you is that the hydrogel is flowing over time, so it's viscoelastic in the sense that if it's in some sense you can bounce on it very quickly, but if you put your hand, it will slowly melt into a puddle. Okay, so the other technique that we use is microrheology, and maybe for those of you who don't know this technique, I'll tell you in a second what it is. We embed the spherical particles in a fluid. So these are two examples. So this is water, just particles in water which perform Brownian motion, and these are the same exact particles in a solution of DNA. So these are a little bit zoomed in because otherwise you don't see them moving. And indeed, this is the point. So the point is that in water they are really jiggling around, and in a solution of DNA they are stuck. There is a little bit of drift, but they are essentially they're not diffusing around, and this is because they are trapped in a meshwork of DNA, which you don't see because there is not enough refractive index for contrast. Essentially, you cannot see the DNA. But you can see the particles, and you don't need to see the DNA. Essentially, you just monitor the particles. And so what we do, we track the position of the particles with an algorithm. So in this case, for instance, I was cutting DNA, so the control, so the full length DNA. So if I have a particle in a very long solution of DNA, this will be very stuck particle, so it will not move a lot. Once it is digested and once the DNA is being cut, the particle moves a lot within the same time window. We measure the mean square displacement, so how much space has the particle traveled in a given time, and then we can convert the mean square displacement using the generalized Stokes-Einstein equation, which I will not go into detail, but essentially it's an inverse, it's a Fourier transform. You can convert the mean square displacement into G prime and G double prime, which are these viscous and elastic components. So essentially we can get the same information from than the bulk rheology, but on much smaller samples. So with microrheology we use typically 3 microliters of sample, and this is useful because DNA is quite expensive, so you want to use small samples. With the bulk rheology we have to use milliliters or hundreds of microliters. Okay, so let's go back to this plot now. So now we can scale it all together. So the curves that I showed you before, we can scale them all together, and something that we learn is that the relaxation time, which is the time where G prime crosses G double prime, so the elastic part, let's say, and the viscous part, they cross over. That is called a relaxation time because it's the time scale at which, or it's the inverse time scale in this case, because it's the frequency in which the system goes from elastic to viscous. It's independent of concentration. But the other thing that because we do this scaling, what we discover is that actually we can scale the plateau here, the elastic plateau altogether, but we have to scale it by a quantity, by the elastic plateau. We have to scale it by different quantities, depending on the concentration. So we take that quantity, so call it G prime plateau. So we are interested in the plateau of the elasticity at high frequency, and we plot it against concentration. And this is the figure that we got. So this is an exponent of 2.5. So as a function of concentration, here in blue, this is a phase separation region where you form droplets, and indeed you don't expect you expect quite low elasticity because essentially it's just made of droplets, the system. But at some point you start to form the gel, and this starts to shoot up, and microrheology and bulk rheology give you more or less the same result of 2.5. Now, the 2.5 is interesting because it's not what you expect. Typically, from fluids, entangled fluids, you expect something that scales like linearly. And the 2.5 was curious, also because other people in the literature didn't find this exponent. So we wanted to understand a little bit better. And so what we did was we did some simulations. So the simulations, this was done by Yair Fosado. So what he did was to essentially fill up a box with a lot of these DNA nanostars, but coarse-grained into beads, into big beads. And then he has 2 methods of computing viscoelasticity. One is to have sort of the equivalent of microrheology. So you embed a massive particle here and you track its motion through this fluid of nanostars. Or the other way is to look at the autocorrelation of the stress tensor. In either way, we get a 2.5 exponent as well from the simulations, either way that you do the green cubo or a sort of a simulated microrheology. Now, this tells me, the fact that we get the same exponent tells me 2 things. Essentially, we didn't change, we didn't change any parameter here. We just got 2.5 straight off the simulations. So it was quite impressive. And so this gave us, one, confidence that the simulations were quite representative of the system, of this experimental system, and 2, the fact that the exponent is, the exponent was right in some sense, because we got it from completely 2 different, microscopically different systems, but somehow macroscopically similar, so the scaling is the same. So we wanted to, then what we thought was, okay, so let's use the simulation to understand a little bit better the system. And so what we did was to do some structural quantification. Let me skip through this. Sorry, maybe this. Yeah, I'll go to this slide. So So one thing that we started to measure was, for instance, so typically in gels, the elasticity is given by the pore size. So typically you look at the pore size and you say, well, it should be 1 over the pore size cube in the elasticity, more or less, just because, just hand wavy argument, just because it's the density of entanglements essentially that give the elasticity to the material. And in this case, so then we started to measure the mesh size in the simulations by having this, by measuring, essentially you throw random points and you fill up a sphere and you ask what is the maximum radius of the sphere that I can fit in that hole, let's say, that contains the random point. And so you can do statistics over it, or you can throw many of these random points in the network and you figure out that this maximum, let's say this mesh size, goes down with concentration. Okay, so the network, so as you increase the concentration of nanostars, the network becomes more packed, and this makes total sense. There is the mesh size decreases. The mesh size decreases, so you expect elasticity to go up. Okay, so this is not surprising. Okay, but the surprising fact is this one. We looked at the loops in the network, and this was the key. This was really what changed everything. So what we did was, well, Yair did, was to come up with a clever algorithm that essentially you look through all the stars in the network, like if they were colloids, and then you track which colloid, which star is connected to which other star. And then you find the minimum loops of those networks. So let's say that this star is part of this loop, and this red loop, you keep only the minimum loops. And we keep only the minimum loops because the assumption is that they are the ones that bear most elasticity. And so if you do a statistics of these loop sizes, you find a very strange finding, which the loop sizes increases with concentration slightly. This is very, if you think about it, this is very counterintuitive, because the mesh, I just told you that the mesh size is decreasing. So what we expected naively is that also the loop size decreased, because if you have a smaller, so you agree with me that this mesh, let's say hole, is bigger than this mesh. So I would say, okay, if I find that this decreases, then also the loop size should decrease. And instead it wasn't. It was increasing with the concentration. And so this was a kind of a conundrum, a bit of difficult to reconcile, until we thought that one way to reconcile the two was because there was network interpenetration. So what we were seeing, and what it can be, we can explain these findings, is that at some point the network decided to form two almost independent networks that thread through each other. So the loops themselves become bigger, but the three-dimensional mesh size becomes smaller. And so we've done a little bit more tests with this interpenetration. For instance, one thing that we did was you can take the simulation and carve a hole, take a sphere in the simulation and carve it out of the simulation, and then you can color the connected components. And what we found is that indeed you can color two independent components, so the green one and the red one. These are two disconnected, disjoint network, which at large enough scales, they will connect again through other paths. But in a small section of the simulation, they are completely disconnected. And the other thing that we did was to go back to the experiments. Said, okay, can we test this experiment? So what we did was to take two nanostars, and two types of nanostars, the design A, design B. We put a green fluorophore on one and a red fluorophore on the other. The sticky ends of A can stick with A, but not with B. And the sticky ends of B can stick with B, but not with A. So what we'll expect is full phase separation. So A goes with A and B goes with B. And indeed, this is what we see at low concentration. But when we increase enough the concentration after a critical point, after a certain point, let's not talk about criticality at the moment, but let's talk about after a threshold. What we see is that there is this emulsion, almost like a red within green, and then if you look closely, you got also green inside red. And so it's and you see a lot of this yellow. So a lot of pixels. You see both the red stars and the green stars, and this means essentially that the two networks are interpenetrated. So you have both green stars and red stars overlapping and forming a co-threading, co-interpenetrated network. Okay, and this is a plot to show that these structures are very stable, so they can resist up to hours or even weeks. We still have this yellow component here. They never fully phase separate like in this case. And we can also do heat cooling experiments, and they're very stable. So then the other thing, then the interpenetration is telling you something. It's telling you that you have so what we started to think, so you have loops and you have interpenetration. If you have loops and you have interpenetration, either you have threading or you have linking. And so we started to look at linking first. So in linking, what happens is, so this is a simulation box where essentially all the stars are made of sticks apart from these one. These loops are bits of stars, of many stars. So for instance, these are two arms, these are another two arms, these are another two arms that are linked. We find that, so we looked at all the loops and we looked at pairwise linking. So and if they're linked, we flag them up. Yeah. So at low concentrations, we don't find a lot of links, but at high concentration, we find that there is a lot of links. So for instance, in these graphs here show that each node is a loop, is a minimum loop, and an edge with another loop is if two loops are connected. So in this case, we get a fully connected network. So all the network is formed of loops that are all interlinked with each other. In this case, there are just a few, and they're not connected to each other. They're kind of clustered. And you find this hierarchical looping, and we also find polycatenates of loops. So loops that kind of linked, and they form a path of linked loops. And so we started to ask, then, is this the reason, then going back to the first slide that I showed, is this the reason for the strange elasticity that we see? And so what we started to look at was, for instance, to correlate the linking with the elasticity. So the first thing was to measure the valence. So for instance, so the valence is how many loops are linked with any one loop, minimum loop. And so we find that as I change this rho is the concentration of stars of nanostars in the solution. What I see is that at the beginning there is kind of, it's very low, but then as I go closer to this rho star, which is the overlap concentration of the stars, so when the stars start to really overlap with each other, I start to see a valence of about 2, between 1 and 2. And 1 essentially means that there is at least one loop that is linked to another one. But this is, if you do 1, it's not enough to create a network. You need 3. So between 2 and 3 here, you start to have a percolating network of loops. Okay, and this scales with an exponent of 1.4. Okay, so on this side you have links, here on this side you have unlinked loops. But so the average number of links valence is 2 at C star, so which represents some kind of transition between a non-percolating and a percolating network of loops, and between 2 and 3 defines the percolation of random graphs. So then what we said is, okay, so let's look only at the bottom here. At rho greater than rho star, or the concentration is greater than the overlap concentration, and the valence is greater than 1, and we expect there, because the percolation of the loops, of these linked loops, to dominate the elasticity. And indeed, so when we plotted G prime. So the G prime is so let's start from the linking. The linking is the red, so this is the same thing that I was showing you before. So the linking, the linking is the number of minimum, so the total linking in the system is the valence, so the number of loops that are linked to any one loop, times the number of loops in the network. So this is my total linking in the network, and this scales as this 1.4 or 1.5 times 1, because there is also the number of time plus 1, because there is also the number of loops in the network. And this scales with about 2.4, 2.5. This is the red curve here. And if I overimpose it with the green Kubo calculation from the simulations, I also get here, so the elasticity, I get the same, exactly the same exponent. But if I instead do an estimation based on the number, on the fact that there is a certain mesh size, I told you about the mesh size that was getting smaller, and let's say that the active elastic components of the network are the number of minimum loops divided by this mesh size cube, then I get this orange curve here, which is at large concentrations is not predictive of the elasticity of the network. So what we concluded is that at low concentrations, the network elasticity is dominated by the mesh size, as in the, let's say, the normal case, like in normal gels. But at the high enough concentrations, when you have this interpenetration, and this interpenetration is really characteristic of our network because it's a limited valence network. Essentially, it's colloids that can make a finite number of contacts. In our case, it's 3. You can make 4 or 5, but 3 is sort of the minimum to form a network. You can do it with a mix of 3 and 2 also as well. But this characteristic is that you can form these loops, okay, and these loops are linked, and we think that it's facilitated by this limited valence network, and the linking between the minimum loops is dominating the elasticity of the network at large concentrations. And so this was actually very recently published in Nature Materials, this story just at the end of last year. Well, actually, it came out in print just last month. So then, when we were doing this work, one other work kind of was a little bit before this, was a work by Francesco Sciortino that looked at the topological nature of liquid-liquid phase transition. And he was looking at low density and high density water networks. These are hydrogen bond networks of simulations of water. And what he said is that when there is this transition, you have a lot of more knots and links in the high density network. And so we saw kind of an analogy here. And so this was a Nature Physics paper in 2022. And so we started to see an analogy here, because in some sense our simulations are sort of macroscopic analogy in some sense of the simulations that also Francesco Sciortino was doing here. So we partnered up with Fausto Martelli at IBM in Bristol, and he had a lot of simulations of amorphous ices. And in particular, he had simulations of atomistic simulations of compression and decompression of amorphous ices. And what you see is that during compression phase, these are all simulations, you get a phase transition here between two different amorphous ices, a low density one and a high density one. So you have, this is a density. This is density as a function of pressure in the simulation. So as you increase the simulation, you have this jump, this continuous jump. And then as you come back, the jump is not here, but it's kind of here. So you have also hysteresis, and so which points to a first order phase transition between two amorphous ices. And no one knew really what was, I mean, how to quantify the difference between, they were calling it just low density and high density ice, but there was no characterization of how the network was different of the hydrogen bond, how the structure of the ice was different, because it's difficult to characterize amorphous structures. And so what we started to look is to use exactly the same algorithms that we use for the DNA nanostars. We looked at the number of loops, for instance, and we looked at, for instance, the number of loops of these minimum loops that were formed during compression. So as you compress, you drastically reduce here the number of loops. As you decompress, you start to come up back here, but it's a little bit difficult to see full recovery. But what was more interesting is that in correspondence to the phase transition, we saw a discontinuous jump of links. So you see less loops, but at the same time, you see a lot of more links. So it was almost like the loops were, instead of having disjoint separate loops in this network of hydrogen bond networks, they were starting to kind of almost like buckle and start to link with each other, have fewer loops, longer loops, and also linked with each other. So we saw the number of links per loop. You see a big jump here exactly at the transition, and then another jump here at the transition back, coming back from the decompression. And as we also saw, although with much less, but we saw also knots. So we also looked at knots, and here you see that it's 0, 0, 0 until the transition. You start to see non-zero presence of knots, and then you go back to 0 at the decompression transition. And so this was a very nice characterization of two amorphosized structures that were just topological. So here essentially we said this is a topological transition between two amorphous sizes because the network of the hydrogen bond network is topologically different. Yeah, you can characterize it with the topological invariants such as knots and links. Okay, so this was published in PRL also this year. Okay, so this was the first part of the story, and now I'll tell you about the second part of the story. Do you want to ask questions? Shall we do it at the end, or do you have pressing questions now? Shall we do it at the end? Okay.
Yes, we will have that question in the end. Yes. Okay.
Okay.
Please go ahead.
Yeah. Okay. Thank you. So this part of the story is about learning knots beyond the topological invariants. So this was work by a very talented master student, Joseph, PhD student Filippo, and then also Yair was involved in this story. So recently, actually, a PhD student joined Georgi. I think some of you might know him because he was in the winter school last December. And we're having a lot of fun with this project. There is I think if you're interested, there is a nice open access article in Physics World called Spot the Knot, and Georgie also came up with a little game that you can play just to understand how difficult it is to kind of identify knots. And well, let me tell you about this story. So the story is essentially, I mean, I'm quite fond of this story just because it kind of has its roots in Edinburgh, where I am at the moment. So these three guys are Peter Guthrie Tait, Lord Kelvin, and James Clerk Maxwell. So they were all contemporary. And Tait, so the road just in front of the institute here is Peter Guthrie Tait Road, and the building where I am at the moment is James Clerk Maxwell Building. And Lord Kelvin was in Glasgow at the same time. So the kind of these three guys were kind of working, were communicating quite a lot together. And at some point, Lord Kelvin was interested in knotted ether because he thought about these vortex knots. So he was thinking about the different elements, different atoms could be represented, could be thought of as knotted ether that cannot be converted one into another. And then at some point, Tait came up with this inventive, this interesting smoke machine where he would essentially, there is a membrane at the back. This is filled with smoke, and if you bump at the back, you start to form rings of smoke. And he will point two machines against each other, and he will show to Lord Kelvin that two smoke rings will bounce into each other. They will not be deformed. They will not change topology. And so he would say, okay, this and this was because of the vorticity of the smoke. And they would say, okay, this is sort of almost like evidence or kind of suggesting the fact that this knotted ether could be actually preserved, almost like representing atoms that cannot be converted into something else. And then James Clerk Maxwell was involved also in kind of these discussions. And so Tait got engrossed, got really interested in kind of coming up with tables of all the possible knots that one could make. And he came up I think in 25 years together with someone else, together with Little, I think, and someone else. He came up with all possible knots up to 10 crossings or something like this. And it took 25 years. And but since then we made a lot of progress in the sense that I think recently the last effort was using computers to come up with tables for 20 crossing knots, and this there is more than 1 billion, I think, knots, something like this. And this was done of the order of hours or days by computers. So this is the change in scale. But then the question, the fundamental question in knot tabulation is always the same: is how do you know that all those knots are different? Okay, because what you want to come up with is, so mathematicians come up with, is topological invariance. So topological invariant is something, as a quantity associated with a curve that does not change under continuous deformation. So if I show you this curve here, I want a quantity that I can compute on this curve that gives me the same answer than the end curve, which is just an unknot. So at the beginning you might say this could be knotted or not knotted. I mean, it's difficult to tell, but eventually you want a quantity that is the same as in the final case. So it gives you the answer that this curve is unknotted. So topological invariants, so one of topological invariant famous one is knot polynomials, for instance. So for instance, the Alexander polynomial, and this Alexander polynomial was used to tell two knots that were thought to be different. These two knots that were predicted by Tate to be different, but actually they are the same. Do I have them? Yeah, they are the same, so they can be deformed one to another. Now they're called the pair copair, and this is just almost like a combinatorial way of assigning essentially numbers to arcs and crossings. You come up with a matrix, and then you get a determinant of the matrix with a variable t, and that is your you get a polynomial in T. And that polynomial is then it can characterize knots, but is not unique. So there are knots that share the same Alexander polynomial. So the open problem exactly is, as I said, is that there are some knots that share the same polynomial. So for instance, here you got the square, granny, and 820 knots. So these are composite knots, and this is a prime knot, and they all share the same polynomial. Another two knots that are very interesting are the Conway and Kinoshita-Terasaka. These ones are 11 crossing knots, and they share the same Alexander, Jones, and Conway polynomials. And interestingly, the Alexander polynomial of these things, which are very complicated, it's 1, which is the same as the Alexander polynomial of the unknot. So this is a very disappointing news for mathematicians. You probably know all this already, but I just want to have, this is the kind of the problem that I'm thinking about. And the other problem that kind of, from my perspective of polymer physics, really I think is quite important, is that there is not a way to determine knot topology from geometry of the embedding. So one for instance, the theory is the Ferry-Milnor theorem that says that if you compute the integral of the curvature of a knot, and if you get something that is less than 4 pi, then the curve must be unknotted. But this is a very weak constraint, because I could have an unknot that is very, very curly, that has a lot of curvature, and is not telling me that curve is unknotted. So it's only a weak constraint on the topology. So then what we started to think is, are there geometric signatures that uniquely identify a knot topology? Okay. So we started to look at different quantities. So we did a lot of simulations of knots. So in this case, for instance, is a trefoil. It's all in here, kind of, and then for instance, you have, for instance, we have a lot of conformations of this trefoil. We looked at the local curvature of the particular embeddings. We looked at the local density. So we slide a sphere along the contour, and we measure how much density of mass there is along that segment. We looked at the 1D right and 3D right, which I'll tell you in a second what they are. So the 1D right and 3D right are defined as follows. So this is the typical Gauss linking number integrand that you will compute if you were to compute the linking between two curves, but here we're using it to compute rise. So if you wanted to compute rise on a curve, which is essentially how much self-crossing there is, you just compute that double integral over itself, over the same curve. And you remove the singularity, removing the singularity, obviously. So the other thing that we did here was to generalize it, to say, okay, let me say that it's a function of the segment position. So segment position is k, and I'm sliding the k, and I'm doing the integral from k minus l to k, and from k to k plus l. So I'm looking only at the riding of two consecutive segments, and this I'm calling the consecutive ride. Oh, so the 1D ride. The 3D write instead is a little bit different, and later on I will call it segment to all write. So this, I can focus on one segment. So let's say segment K, and then I'm integrating from K minus L to K plus L, and then I'm integrating over the rest of the contour, this integrand here. And this is telling me how much the segment K is writhing, is entangled with all the rest of the segments of the polymer. And so in this particular simulation, this particular snapshot, then what I see is that the local curvature is all over the place. I see a lot of bumps here. It's not very easy to see any pattern. Local density, I start to see two peaks here, which represent a little bit kind of the edges here, these two beads here. Over there are two beads in cyan and purple. 1D right, I see a massive single peak here in this region, probably because of this twist. And the 3D right, I see two peaks here, okay, which also correspond to these two beads, which represent the end of the knots. And so we started to see, okay, these are patterns. We could maybe, you can, by eye, a human can start to interpret these patterns. But if you really want to do pattern interpretation, what you have to do, you can use machine learning to do it. Okay, so then this is what we did, was to take knots and do long simulations. So this is a long simulation. Now this is going back and forth, but this is it's just a little window of a very long simulation of a knot. I think this is a Conway knot, and we take snapshots at distant times, so to have independent conformations, and then we compute for completeness. We compute this. We call it a segment to segment, right? And this is a heat map, is a two-dimensional map, is calculated on segment X and segment Y. So every two segments, every two beads of this coarse-grained polymer will have a point on this map. And regions of bright regions represent that 2 points are very entangled, and low regions represent that two beads are not very entangled. So this is the segment to segment, right? And then we can do, if you do the integral or the sum over rows, for instance, then you get the segment to all right, okay? Which is just the integral over this segment to segment, right? So we start from these patterns, which are the same that I showed you before, so the segment to all, and we input it into a feed-forward neural network. So what we're doing here, we're training the network, so this is a supervised learning. We show a lot of patterns from unknots, from trefoil up to 5-2, okay, 5-2 knot, and we train the network to recognize the patterns and to classify them into these simplest 5 knots. Okay, and what we get is the following. So if we train the network with the XYZ coordinates of the beads, we get a decent result. So this is something also other people have done before. So the feed-forward neural network gets to 80% accuracy, which is pretty good, and a confusion matrix. A confusion matrix that shows a little bit of confusion here around the 5-2, 5-1, 5-2. But when we use this STA write, we shoot up to 99.9%, and the confusion matrix goes completely diagonal. So this is a brilliant indication, a great indication that the write, this segment to all write, contains more easier information for the network to understand the knot topology, other than the embedding itself, other than the XYZ coordinates. So then we said, okay, this works very well. Let's challenge the network with knots that share the same Alexander polynomial. So we did the simulations of the Granny, the square, and the 820 XYZ coordinates. If we train the network with the the XYZ coordinates of the knots, it works okay, but the STA write is 99.9%. More strikingly, we did the Conway and Kinoshita-Terasaka and the unknot, and this is not. These two are not distinguishable by the XYZ coordinates. So the neural networks could not distinguish Conway and Kinoshita-Terasaka knots. But the STA write is, again, fully diagonal, 99.9%. So neural networks trained with this segment to all write can distinguish knots with identical knot polynomials. We then did, what we did was to look at higher crossings. So we did 6 crossings, 7 crossings, 8 crossings, up to 10 crossings actually. and accuracy goes down for the XYZ a lot, but for the STA right, it remains pretty high to 85%. But then we started to realize that they was starting to get confused here. And so we looked more in detail about these knots here. I think it was 5.1 and 7.2. And so we started to see that the STA right is quite similar patterns of these two knots. And so we decided to upgrade, let's say, our feature input and use the STS matrix, because the STA essentially is just a projection, is a sum over the rows of this matrix, which maybe contains more information, and it does contain more information. And so if we start to train with the STS right, we're not confusing these knots anymore, and we go back to the 99.8% accuracy, even for seven crossing knots. Now, there is a lot of knots with same Alexander polynomial. These are all the knots with the same Alexander polynomial up to 10 crossings, and plus all the mutants will have the same skin polynomials. But still, even if we train our network up to 250 knots, which is up to all the knots of the Tate table with 10 crossings, we still get 94%, 95% accuracy actually, which is pretty impressive. And so we started to ask why, and here I'm really kind of going to the end, so 1 or 2 slides and I'm finished. So now Georgie, what he's doing is to try to figure out why the network works so well with Wright. And what he's figuring out is whether there is, so the question is whether there is a connection with order 2 Vasiliev invariants. So in the sense of, is the network trying to compute the topological invariants from the Wright? And this is what we hope to understand. So what he's doing here, for instance, is comparing the true value of the Vasiliev invariant from the, let's say, the distribution of the measure of the Vasiliev invariant from our simulations, and we are pretty close to the true value. Okay, so but the problem is that the order 2 Vasiliev invariant cannot distinguish all those knots, okay, all the knots up to 10 crossings. So, and also the Conway and Kinoshita-Terasaka, they require a much higher order Vasiliev invariant, which is this. A Vasiliev invariant essentially is a combination of this Wright integrand that I was showing you before. And so what Georgie is thinking right now is that perhaps the network is doing something else. Is learning from the Wright. Is looking at the Wright and the number of peaks. And so this is a final slide. This is showing you, for instance, the XYZ coordinate of a knot. This is why the quality is not too good, because it's really recent results. Is that Georgie started to look at the saliency of this is the weights matrix of the neural network, and trying to figure out where the network is paying attention to make a certain decision. And what he's figuring out is that he's not actually looking at the regions of maximum entanglement, but he's looking around them. So maybe he's looking at, I don't know, maybe he's looking at, we're still trying to figure out what the network is actually doing. So that's the answer. Okay, so I'll draw two conclusions. So I showed you a story about limited valence networks that display topological viscoelasticity with DNA nanostar systems done both in simulations and experiments. And we showed you the simulations and experiments match pretty well, and that it's quite convincing that they form this interpenetrated network structure where there are loops that are interlinked with each other. And we can also explain this topological transition between amorphous ices. And then I showed you the second part of the story on machine learning that seems to be able to learn and to classify very well also difficult knots that share many topological invariants. And we hope in the future to understand better how these networks do it in such a way that perhaps we can come up with better topological invariants for knot classification. I'm going to stop here. Thank you, and just acknowledge these guys, which were the ones doing most of this work, and thank you for your attention.